this might, oh, come on, yeah. So what if we don't know how the brain can do something, yet the machine can do it? Then that's, even better. For example, language, right? We have essentially no models of how language works in computational neuroscience. I mean, it was people largely didn't study it because it's very hard to come up with neural data for language. And now we have machines that can produce we always had machines that can produce language, but we have machines that can produce convincingly language. Now we can then study these machines and get some hypothesis about how the brain generates language, right? The machines are, once again, not necessarily doing it in the same way that we're doing it, but they can give us hypothesis. So, oops, oh, okay. I guess I was going to talk about that quote. And then if we look at the bottom left, you have the case where we have some ideas about how the brain does something, but we don't know how machines can do it yet, right? And then this is a clear case where the AI can learn from the brain, right? And AI has already learned from the brain in some major ways, and then you can continue in this direction if we happen to be in that quadrant, right? And finally, if there's something that the brain can do, but we don't know how it does it, and we don't know how to make machines that can do it, then that's a very exciting thing, right? It's a really a research frontier. Okay, well, I need to move this a little bit closer. Yeah, so that's an exciting challenge, right? So essentially, there is something special about the brain and AI because first, AI and the brain are often focusing on similar things, right? Artificial intelligence, that is to some extent, but now it has grown more than that, but to some extent it is to try to mimic and surpass human intelligence, right? So that is the function. There's a lot of similarity there. And because the underlying structure, right, the artificial neural networks use neurons and the brain use neurons, so there's a lot of comparison that can be made at the lower level as well. So that's why these two things have a special relationship, right? AI can be used for many, many scientific endeavors, can be used for data analysis, but when it comes to modeling the brain, it has a special status. Any question about this? Okay, so this is just a generic argument that there's always something to learn. It doesn't, what this is not saying is that AI is necessarily a good model of the brain, right? So we shouldn't confuse these two statements. Now I want to show you first an example from our own work. I mean, I will be focusing on our own work, but just as examples, a case where artificial neural networks can be remarkable model of the brain. Okay, there can be a remarkable convergence between the two. So many of you probably heard of convergent evolution, right? So this is the phenomena where animals that are separated very far apart in evolution history, right, converge onto similar solutions to something, right? For example, dolphin and shark are separated by many, many million years of evolution, yet they have very similar body shape, right? And that tells you something fundamental about their body shape, right? It's not coincidental. There's enormous evolutionary pressure to get to that body shape in that environment. So every time you see something where there's a convergent evolution, right, that should tell you something about the strong evolution pressure that is applied to the system, right? And it can help you understand the system better. So one thing that, another example of convergent evolution in neuroscience is the olfactory system that I will tell you about. So most people who study sensory systems focus on vision and auditory systems, But the olfactory system is fascinating because it's both, it's actually quite similar and it's quite conserved. So this essentially, it's a 3-layer neural network that starts with olfactory receptor neurons. So these are cells in your nervous system that essentially lie in effectively the animal's nose, right? This is a fruit fly. It doesn't have nose, but these neurons are at the forefront of the whole machinery, and they're receiving chemicals in the air, and they're sensing that, and then they project to a second layer of neurons that you don't have to remember the names, but they're called projection neurons, and then they go to another layer called Kenyan cells. Once again, you don't have to remember the name, and that's pretty much it. So there are these 3 layers of processing. And I will tell you a little bit more about how they're actually connected. And I promise you, even if you're not a neuroscientist, you would be able to understand it. So you start with these input neurons, right, these sensory neurons, and there are different types of them because you need different neurons that essentially prefer different types of chemicals, right? Some neurons are specifically activated by CO2. Some neurons are specifically activated by, for example, fox urine, right? This is, some neurons are really selective, which means they really prefer fox urine in mouse. So you have these different types of neurons, and for each type, there are multiple of them. Okay, so in the fruit fly, there are 50 types of neurons and 10 neurons per type. Okay. Oh, by the way, I should have mentioned in the beginning. Please interrupt me anytime. I'm happy to just keep this as a discussion, and I'll watch time myself to make sure that I don't get derailed too much. So it's all good. So you have these 50 types of neurons and 10 neurons each. And then they go through a very strong compression, which means the number of neurons go down substantially. And it goes to about 50 neurons. It's a little bit more complex than that, but let's for simplicity, let's say it goes to 50 neurons. And what is remarkable here is that the connections are very, very precise. Each neuron in this compression layer only receives connections from one type of neurons. Okay, so if you think about this as humans, right? So you have this is actually pretty intuitive. There is no reason to think about it as humans. So you have this very organized, very, very organized activity, right? That goes from neurons of the same type to one neuron here. But then what is really strange is the system seems to just throw that all away, right? It will go from this compression layer to then an expansion layer that is much larger, that has about 2,500 neurons in the fruit fly. And the connection from this compression layer to this expansion layer is largely random, which means there's very little structure in this, right? So you go from a very organized connectivity to a largely random connectivity from the second to the third layer. And because it's random connectivity, you can actually summarize it with just more or less just a single number, right? And again, it's a little bit more complex than that. But you can more or less summarize it by a single number, which is how many connections does each expansion layer neuron receives from the previous layer, right? So if you know that, then you can essentially generate the random connectivity yourself. And people know this number in these systems. And it's about six connections, okay? So with this organized connectivity and then random connectivity in the second layer, where each neuron receives about 7 connections from the previous layer. So that is the more or less the fruit fly olfactory system. And like I said, what is remarkable is that there's convergent evolution between the fruit fly and the mice that are separated by 500 million years of evolution. Mice has a very similar structure, except with larger network. So it has about, it has about 5 million neurons here, and it has about 1,000 neurons here and 1 million neurons here. Anybody has question about this architecture? Yes. Does it mean in each fly is different? Okay. That's a good question. I actually don't know, because I'm not sure people did this across flies. So I honestly don't know. But when I say I run them, I mean, if you are to draw a random matrix, right, you're just saying, well, for example, which neuron should, this neuron should receive, for example, 6 connections from the previous layer, right? I'll just randomly pick 6 neurons from these 50 neurons, right? And then you'll be able to draw a matrix like this, draw connections like this. And then you look at the real connection from the fruit fly, and you can't tell the difference between the two. So you can do statistical tests, and you wouldn't be able to tell how they're different. So that's what I mean by random. Okay, and again, if you're an expert, it's a little bit more nuanced than this. But this is a first order approximation. Yes.
You say it's a convergence between the mouse and the fly, but it can also be that the common ancestor had the same factors and then it was divergent.
That's right. So it could also be preserved across evolution, right? So I'm not an olfaction expert, and I don't think people have mapped out the olfactory circuit across all these different, all these species on the evolution tree. So I can't quite. Okay, let me just say it's either highly conserved across evolution or it's convergent evolution. But in both cases, it suggests that this architecture, this kind of organization is important. Yeah. Okay. So now our question is, can we have a machine reproduce that, right? So this organization is a result of evolution. It's actually not a result of development, of learning. It's largely just hardwired through our gene, through their gene. And our question is, can we replay the process of evolution in machines and recover this connectivity? And if we can do that, then we might be learning something about the evolutionary pressure that these animals experienced, right? So what we did, so together with Peter Wang, Richard Exo, and Larry Abbott, what we did is design a very, very simple task to do this, okay? So I won't explain you the details of the task, but I will explain you one idea about this task. So for vision, if you want to have a neural network that can do some visual task, right? One classical thing to do is object classification. You have a bunch of images. In each image, you have an object, and the network has to say, is this a cat, is this a dog, is this a computer, right? Now, how do you do that for olfaction? It's much harder because we don't have a data set where we know how the neurons are activated at the first layer, given a peach, given an apple, so it's very hard to do. So instead, what we did is that we built a very simple, embarrassingly simple data set based on just one principle about olfaction, okay? So if you take 2 images of a cat and you average them, what do you get? You just average the 2 pictures. What do you get? It's not a cat anymore, right? And if you take 1 million images of cats and you average them, you're probably just going to get a bunch of noise, right? The same thing is not true for affection, right? If you take 2 apples, each of them smell like apple, you combine this smell together, they would still smell like apple, right? And if you combine 1 million apples together, yet they would still smell like apple, right? So there is something about affection that distinguishes it from vision, from audition. And this is a property that we used to design a simple artificial task for affection. And we trained the network to do this task. Okay? And if you talk to affection expert, they would say, well, this is missing all the complexity, all the nuance that we know about real affection. And they would be absolutely correct, right? And so, but this only makes what I will show you next perhaps more surprising. So we train the network on this task, and what I will show you next is a little animation that shows you the connections and part of this artificial networks, where I'm only showing you 3 types of input neurons, all the 50 neurons in the compression layer and 5 expansion layer neurons. So where we start an artificial neural networks that has the same size as a fruit fly olfactory system, but completely different connections in the beginning, right? As you can see, in the beginning, all the connections exist. So it's all-to-all connectivity. And what I will show you next is what happens during essentially the training of this network on this task. So you can think of it as a way of replaying the evolution, but in the machine. And what you can see is very quickly structure emerge in this network. And you can see there is a remarkable resemblance between the artificial network and this schematic of a biological network. In that each compression layer neuron, when it receives input primarily from the same type of input neurons, right? And each expansion layer neuron receives connections from a small subset of the compression layer neurons, right? And in fact, you can count how many connections each expansion layer neuron receives, right? And we can do this together. Okay, so let's, if we focus on the green line, this is about 1, about 2, 3. And this is a bit tricky, right? Maybe it depends on what you think. So we can do this in a much more accurate fashion, but just to give you a sense, 1, 2, 3, 4, 5, 6, 7, right? 7 or maybe 9, depending on whether or not you count the weak ones. Now you can do this in a much more accurate and quantitative way by just actually plotting the distribution of connection weight and setting thresholds. So we have done many different things here. But yes. From the convolutional network used in vision, from a stacked hourglass. Yeah, so it's, for example, this is not convolution, it's not trained to reconstruct the input, so there's many differences, yes. But there is, I guess, the similarity is that there is compression and expansion, but there are many differences, yeah. So, long story short, we can quantify this, and you can, happy to see that, so you can check out the details in the paper. So you can quantify how many connections each neuron receives from the previous layer. And it turns out you get a remarkable match between the brain and the machine. So this is a histogram that shows you the distribution, right, of connections that each neuron receives, right, the input degree to each neuron. And people have very recently mapped out the almost a full connectivity in a single fruit fly. So this is work done by Janelia Research Campus, and it has an average of 6.4, right, out of 0 to 50. And in our network, we get an average of 5. So this really should only be a start, right? This alone is not an understanding. It's just a demonstration that you can get, in some situations, a remarkable convergence between the brain and machine. And in this case, it's actually quantitative. And once you have that, then you can say, well, why does this happen, right? You can try to change it, change the different constraint that you are giving the network. You can change the task that it's supposed to do. You can change the architecture of the network. You can change many things until you feel like you understand why this is happening, right? And I won't have time to talk about that today, but I just want to leave you with this impression that sometimes these neural networks can be quantitatively good models for the brain. Any question about this part before I move on to the next part? Yes?
Yes, actually the question is then how much actually this is surprising that this converge to each other based on that neural networks, the architecture with activation, function, and all this gradient. So it's basically based also on the how the brain works. So I mean like this neuron structure, you have the synapses and dendrites and cells. So is it be surprising that this converge to the same things?
Yeah, so I think it's, so all the things that we have put in here is a task that we think is relevant for affection, a network that has the same size as a fruit fly, and for people who are neuroscientists, the connections from one layer to another respect the biological fact that they are so-called excitatory, which means each neuron can only make the activity of the downstream neuron higher. But many other networks have that. And so people have built many, many other networks that follow these principles. But I don't think I've seen anything where people can show a quantitative match between the connectivity of network, artificial network, and biological network. And for one reason is that people don't even know the connectivity of biological network in most cases. So this is a very unusual case where you can summarize the connectivity in such a simple fashion. There are maybe like a couple other cases in fruit fly where people can say there is a convergence between the machine and the brain. But yeah, I would say this is pretty unusual. Yes?
Yeah, it's certainly impressive, I must say, but is that not a risk that you tweak your system towards the biological knowledge?
Right.
And maybe if I do this 100 times, then I get
Yes.
8 times another
Yes. So this is always a concern, right? Like that happens when you already know what's going on in biology, and then who knows, maybe I tried a million things until we get to this point. So so this is a great thing, and I want to spend a bit of time talking about this because this is a general thing as well. But first, anecdotally, we get this result the second day of starting the project. So we spent 2 years publishing, following up and doing the work because we were so surprised that we got this in the beginning, and we thought, okay, we have to be super careful and try many, many things. But the general answer to this, I think, is we cannot stop when we see a match, right? We cannot just congratulate ourselves at a match and just publish that. We have to understand more deeply the reason and the limit. So for example, if you get one hyperparameter setting where you can have a match between the brain and the machine, then between the machine and the brain, you should try many different other hyperparameter settings until you see when it breaks, when the match breaks down. So, for example, in this paper, our supplementary figures are largely that. We just try many different things, and we show when it's a consistent, when you get this, and when it breaks down. And so it turns out for most things that are uninteresting, it doesn't change it. For example, the most trivial thing is random seed, right? When you train a network, you have some random seed, and that doesn't matter at all. And there's some other things, like, for example, the amount of noise you give to the system. So we have tried many things like that, and none of them matters, but there are things that do matter, and so we talk about that in the paper. And one of the things that matter is this fact that the connections are excitatory, or excitations, and that would actually give you some insight about why the system is organized this way. Like, once you know, oh, it's because that it's excitation, it's only with excitation that you get this would help you understand the system better. Yeah. Yes, Tavik.
Robert, I just wanted to ask a variation on that question.
Yeah.
Just to connect it to what a lot of other colleagues do. So some colleagues are working on genetics or
Yeah.
oncology or other clinical topics. And so the question would be, when you don't know the structure, the biological structure, how can these kinds of tools aid in discovery?
Yes.
And I guess some of the answer is constraints. So what constraints would you recommend that we
Yeah.
would use to help us aid in discovery of the structure?
Yes. So that's a great question. I don't have actual recommendations yet, but this is something we're working on. So I think the best thing as a whole community we can do is really to pull data together from many different labs. So when we build models, we can try to explain results not from one lab, but from, say, 100 labs. Right? And if you can now build a model, and you can identify the constraints that allow the model to describe and match data from 100 labs, then there's a lot of hope that then it can generalize and make predictions to results from 1,000 labs. So I think that's my general kind of answer to that, and it's not an actual recommendation, but my actual recommendation would be it's great to work on data sharing platforms and stuff like that. It's
I
yeah.
guess I was asking more in terms of regularization of the network, or
So,
in terms of what
yeah.
kinds of cost functions would you recommend?
Yeah.
And how would you validate, how would you constrain the number of hypotheses that would be generated for discovery? So in case you didn't know what the fruit fly kind of connectivity structure looked like, how would you have approached this problem?
Yes. So if we didn't know at all, it might be harder. So what we will do, what we can do is the following, which is something we ended up doing anyway, which is essentially do a search in the whole space. So here I showed you a network where it has the same size as a fruit fly, right? It has compression, expansion, but it doesn't have to be, right? You can train a network that is larger than this, that is smaller than this, that is deeper than this, that is shallower than this. And so we did all of that. And what we found is this is actually quite almost an optimal structure. And I don't say that word lightly. And so what we mean by optimal here is getting rid of any neuron here would decrease the accuracy. I mean, you can get rid of the expansion layer neurons, and they would just decrease the accuracy gradually. 2,500, that's more or less an arbitrary number. But if you decrease the compression layer neurons, that would decrease the accuracy very quickly. If you reduce a layer, it would decrease the accuracy dramatically. If you add a layer, it doesn't improve the accuracy. If you add more neurons, it doesn't improve the accuracy. So one thing you can presumably do then is to explore this entire space and then try to find the optimal network in this entire space, and that could give you some hint at what the architecture in the brain can be. And by the way, here I talked about mesh to fruit fly, and we do make predictions about the structure in every single animal, right? Because we can train this network with different size. And like I mentioned earlier, the mouse has a larger network with 5 million neurons here, 1,000 neurons here. While we cannot train a network that big, we can train a network that is, for example, 10 times larger than this. And then we can quantify, for example, the connectivity here. Oh, by the way, no matter how big the network is, the overall architecture always come out the same. But the quantitative, we can also make quantitative prediction about the input connections that go into this expansion, each expansion layer neuron. And that we were able to make a prediction, right, that essentially it's a function between the number, it's a function between the input degree to each neuron and the number of input, the number of different types of input neurons. And it's a power law, which allows us to essentially make a prediction, and it could potentially be, I mean, honestly, it's likely wrong. I mean, there are many things that maybe we didn't account for, but yes, we can make predictions as well. Yes?
Yeah, I have a question about the machine evolution of olfactory structure. I'm
Yeah.
curious about what kind of information for input, like the different chemical structure. Like,
Yeah.
I think two example about smell apple. So for the smell apple, I think they be composed of different kind of
Yeah.
molecules.
Yes.
So you put different kind of molecules.
So this is a part that we glossed over, that this for olfaction expert, I mean, Richard here is a world-leading olfaction expert, but for many olfaction expert, this is, once again, embarrassingly simple, right? But our goal is not to build a data set that is so realistic. Our goal is to find the minimal condition you need to achieve a match between the brain and the machine, right? And we want to find the necessary and the sufficient condition, right? So if anything, I think having a simple data set that doesn't capture some of the realism and showing that still works, I think that's even more interesting than having a very realistic data set. Okay, I'm happy to come back to this, but okay. Yeah, so the input data, okay, maybe I'll just briefly explain this. So if you plot the activity of these input neurons, so there are 50 types and 10 neurons per type. So within each type, the activity is similar. So it's essentially a 50-dimensional space. And then you can plot each point as a dot. So each order as a dot, and they would occupy somewhere in this space, and all you need to do is a classification task, where essentially there are some prototypes that are described by these squares, and they're just randomly placed, and then an order is assigned to its nearest prototype, the class of its nearest prototype. So it's a very, very simple task, right? Yeah. Okay, I'm happy to come back to this afterwards if people still have questions, but I do want to make another point, and by telling you another story. So I showed you that one example that machines can be very helpful to study the brain because they can be quantitatively good models. Right? This doesn't always happen. but sometimes it does. Another thing that is quite helpful about these neural networks is that it can help us ask the why question about the brains in machines, and I'll explain what that means. So some of you, if you're a neuroscientist, and I'm sure then you have heard of this, that there are some neurons, some brain regions, some regions in our brain that are that really like face. Okay, so the technical term is they're face selective. And practically what that means is if you show, for example, a human lots of images, right? A face, an object, some place, some food, right? Anything you can think of, then only the images that contain face would activate these neurons and would activate them strongly, right? And all other images would activate them very weakly, right? So this is a very special thing about the brain that was discovered, at least through fMRI, by my colleague Nancy Kenwisher and others. And for 25 years, people know that this region exists in our brain, but what is unclear is why. Why does it exist, right? It didn't have to exist. You could imagine the brain without faces, right? In fact, if you train just a generic artificial network to do object recognition, it can still recognize faces to some extent, and yet it doesn't have a face area, right? So why is this happening in the brain? So this is an example where we know that the regions are there, we know what the neurons are doing, but we don't know why they're there in the first place. Okay, and so this is just one example of specialization in the brain, which means you have some brain regions that are specialized to do something. But our brains are full of this, right? We have many different brain regions that do different things. So why is that? And one hypothesis, right, you can get is by just observing a company, right? A company can have different departments, right? It can have a finance department, a human resource department, and it has that because that is efficient, right? It's better to have that rather than having everyone do a little bit about finance, a little bit about human resource, right? So a hypothesis is if you have an organization of people and then you have so many things you need to do, it's better to have these specialized department, right, that focus on individual task. So I was essentially inspired by this analogy and wanted to look at what happens if artificial neural networks has to do many things, and especially many cognitive things that animals are known to do, right? If our previous hypothesis is correct, then we may see that this neural network would specialize into different different modules, right? Just like in a company, it would specialize into different departments. So just to visualize what I described, if you have a group of neurons and you train them to do, for example, two things, like finance and HR, but it can be just any two tasks. So on one extreme, you might have groups of neurons that are specialized to individual tasks, right? Or an extreme alternative is each neuron is doing a little bit of everything, right? So this is kind of like a early small PhD lab where everyone is doing a little bit of everything, purchasing, right, buying computers, stuff like that, right? Until you start to specialize and you have, I don't think PhD labs ever specialize. So, and then there are other hypotheses, but today we will just focus on this. So just to remind, remind you, we have a recurrent neural network, which really just means the neurons are connected to one another. And then we train it on many cognitive tasks that neuroscientists have studied. So these are relatively simple tasks about decision making or about memory. You have to remember something in your mind for a second, or you're looking at a bunch of dots and you have to decide whether they're going left or going right. So tasks like that and variants of tasks like that. So we found 20 tasks from the literature, and then we trained a network to do all of them, right? And now we can test whether or not in this network you have these, clusters that emerge, which is more like the department, right? The departments in a company where it's like this, which is more like a small research lab, right? And so this is what we found. So before showing you what we found, so I will show you a plot where on the x-axis we have each neuron in the model, and on the y-axis we have the task, and using the color I will show you how each neuron is involved in each task. So it's very similar to our previous schematic where we would show how each neuron is engaged in each task, but instead we would show them across all the tasks. Okay, and if it's completely mixed, then you should see each neuron, which corresponds to a column, would have some white color across all the tasks, right? And if it's extremely modular, then you should see some group of neurons that are involved in one task, some other group of neurons involved in another task, and so on. And what we see is neither. What we see is somewhere in between, that we find some groups of neurons, for example, this group of neuron, that is involved in this subset of tasks. So it's involved in not just one task, but also not all the tasks. So it's involved in a subset of tasks. And this is a common theme, that you find these clusters of neurons, these modules that are each involved in a subset of tasks. So what does this mean? It means a network, we're given all these tasks that it has to do, but it realized that these tasks are not independent. There's some common computation underlying some subset of tasks. And then it was able to develop these modules that serve these shared computation. And then when you need to solve a task, you don't use just a single module. You would use a combination of modules. For example, if you're solving this task, you would use this module and you would use this module, okay? And so each module is supporting a common computation across a subset of tasks, and each task would typically engage multiple modules flexibly. Just to give you another analogy, so this is almost like the network, through training, developed a toolkit, and it can use it to do many tasks, where each task would involve multiple tools. So this is an example where we can provide a tentative answer to why the brain have these specialized regions, right? In this neural network, it is just from the pressure of doing multiple things that you would get specialized modules that are focused on common computations. Okay? So I think I'll just very briefly just say these are two examples of more of a success story, but there are still many problems with using artificial neural networks as models of the brain. that I won't have time to talk about, and I also already covered some of that yesterday if you were there. But that shouldn't discourage us. So there's really no fundamental barrier between these artificial neural networks used in AI and the more classical neural network models that people used to build before deep learning exists, right? And we should be open-minded and not like fall into camps, right? Sometimes people feel like, oh, this is like a school, that's all we do. We have to do artificial neural networks and train them with a fancy machine learning algorithm. It doesn't have to be the case, right? I think the best way is just to start from the research question. What is a question that we want to study? And then we just use whatever tools are best for studying that questions. And if it involves changing our tools and fixing it, then so be it, right? So we don't have to be purist. So with that, I'll end and thank people in my lab. Sorry, it's just, this is widely, I've shown this yesterday. So I'll thank people in my lab and our funding. Thank you. Yeah.
So we're going to briefly open the floor to questions, and only have a few questions, and then we'll proceed to the room. Any questions? For Robert?
Okay.
Yeah, thank you.
Yeah.
Yes, thank you. First of all, thank you for a really interesting presentation. I actually have a question. Maybe there was like a follow-up of this discussion about is the
Yeah.
structure evolutionary preserve or is like
Yeah.
converged from different species? Actually,
Yeah.
I was thinking, can you maybe also prove it via these simulations in a way that, like you said, that you found the structure like on second day, right, of the experiments?
Yeah.
So, and then let's say if this, I think it's K number, right? What was the average number of connections or something you showed on this slide? Yeah. So let's say if this K number is like something you always converge to this, that's probably can be proved like for the convergence.
Yeah.
But if it's just unique situation, let's say when it's let's say happens just well in some parameter space and others there can be different solutions, that might be more for the evolutionary preserve, right? Structure. So what do you think?
Yeah. I didn't quite understand. Can you say it again?
So let's say you said that the structure when you train the network, right?
Yeah.
So that the K number
Yeah.
came with the parameters at the second day of the experiment.
Yeah.
So and then this you said that you could find some boundaries where this doesn't work, right?
Yeah.
So but then the question is it something unique when it doesn't work, or it just subset of the parameters? So say you have all the hyperparameters right in the network. So in some subset you will fall to this K number like in real life.
So
And
I see.
but could you use it to prove the hypothesis? Let's say, is it like evolutionary preserved structure, or is actually converge to this structure via different passes?
Okay, so I don't know if we can prove whether it's evolutionary conserved or it's convergent evolution, but I can tell you what happens when we do these hyperparameter sweeping. So there are different, essentially, there are interesting hyperparameters and there are less interesting hyperparameters, right? So you want your results to be robust to the less interesting ones, and it might be still robust to the interesting ones or might not. And I'll give you an example of what's a not so interesting hyperparameter. For example, when you train a neural network like this, for people, many people probably have experienced, if you have to set a learning rate, right? And this is just one of this hyperparameter that is common to machine learning, but usually you don't expect that to change your result too much, right? And if it's very sensitive to that, it's bad. And so, for example, we show that it's not sensitive to that. We can vary it in a range of like 100 or so, and it doesn't change the result. And similarly, about the random seed, Now there are some more interesting hyperparameters. For example, people have hypothesized that you get this structure of input then compression for noise reduction. So this is like a leading hypothesis for explaining why this is the case, right? And so this is something that we can vary. It's an interesting hyperparameter because we can vary the amount of sensory noise that is presented in these input neurons. And it turns out, in our case, you can vary them from 0 to pretty high, and it doesn't change the result at all. So this is a case where the result is robust to this hyperparameter, and it tells us something. It argues against a leading hypothesis. And then there are situations where if you change it, it changes the result. For example, if you increase the number of expansion layer neurons, or if you increase the number of input types from 50 to 100, this would go from 5 to something like 10. And this also shows that the number 5 is non-trivial, right? You have to get it with the right value, but we are using the value that is same as fruit fly. And then so there are different types of hyperparameters that you can look at, and our general approach is just to look at as many as we can, and we only report things when they're robust to the not interesting ones, and we report how they depend on the interesting ones.
Any other questions? So maybe just for the sake of rounding this whole discussion off and connecting it to the broader biological community. So this modularity thing, Robert, could we go a few slides later to this module thing? I guess the one question one might ask is that, what kinds of features does a biological system need to have, whether it's a genetic system, whether it's a heterogeneous population of tumor cells, for this kind of modularity to emerge?
So I think this modularity, actually there are a couple recent papers on this topic, and the people have followed up on this. So there's some technicality with how you define modules. For example, if you have neurons versus tumors, you may define them differently. But the general principle here is, if you have a system that is quite optimized to do many different things, and these different things can have some shared computation, then the system would tend to essentially extract these shared computation and specialize for them, right? So it's kind of like how we have different organs in the body because they specialize for different tasks.
A lot of gene systems.
Yes.
Proteins.
Yes.
All right, so with that, I would like to thank Robert, and I'd like to remind everyone that as a community, one big goal of this project is for us to also get to know each other and communicate with each other. And for that reason, we organize borrows after the talk. And I know everyone is eager to get home, but if possible, if you could stay back for just a single drink and just try. The idea is not just to exchange science, but to also discuss it as a community. So with that, I thank Robert again, and I thank all of you